nominated as a top young researcher to meet the Nobel laureates in 61st Lindon Nobel Laureates Meeting 2011 in Germany. He is also known for his in-depth work on several ethnic groups of South Asia, including Himalayan, Andaman, Siddhi, Roma, and Parsis. Based on his and his team's research on Roma, late Srimati Sushma Swaraj has called them the children of India in the year of 2016. He was also invited by National Geographic USA for presentations and panel discussions on who we are in National Geographic Symposium in June 2018. He is among the list of 10 Indians to watch out for in 2019 by Swarajya magazine. On October 2019, he was also felicitated by Honorable Home Minister, Government of India for his outstanding work in Indian prehistory. He has also received the Top Researcher Award to 2020 in the Institute of Science, BHU, and he is also writing regularly editorial columns in the largest Hindi daily, Dainik Jagran, on COVID-19 and India. Sir, please.
Can we switch off this light? Okay, so everyone, how is Josh? I'm very sorry coming in between your great lunch and but I think I'll be very quick and finish the talk. So just please give your 20-25 minutes. We'll be able to deliver something. So I'm grateful to the organizer as well as Dr. Ankit ji for inviting me here. It's a great opportunity. And sir who are sitting in dais, Professor Kumar sir, who has done a lot of work and who has shown some of his work here, and chairperson who is chairing the session. So, you know, in forensics, the development of the technology is quite crucial. So initially I started to work in the CCMB Hyderabad under Dr. Lalji Singh, who was the in charge of the lab. So there I used to learn about different cases, but there was a thing that because the problem in forensics is like you cannot change very quickly because you need a huge amount of databases, and these databases takes time. So without the databases, developing the technology, using this developed technology is completely impossible. So what we do, we produce the database, we make the database for the people, for the forensics, and then introduce new things, introduce new technologies, and tell them that this technology could deliver much better answers of your question. So here I'll be bringing about the Y chromosome, because whenever you read some forensic cases, there are more than 90% cases in India or even the world has been done by the males. So the development of Y chromosome related, ancestry related panels and Y chromosome related markers are very important to move forward. So here I'll be presenting about the technologies which should be used to get better result, and also some of the cases, some of the population genetic related cases where we see that how we have solved the problems. So there is a very simple differences between population genetics and forensics. You need large number of individuals for population genetics because it's related with the population. But forensic is individual specific. We need to know information about a single person. But why population genetics is important? To get the information about an individual, you would be able to know further. So in population genetics, common variants are needed. There are a lot number of mutations, but in forensics, rare variants, very specific kind of mutations are needed to understand the ancestry of an individual. Population genetics rely on enough amount of DNA. You go to the field, draw some 5 milliliter of blood, have kilos of DNA, and then you can do whatever you want. But in forensics, as you know, as even sir has presented, we get only less amount of DNA. And for population genetics, there are hundreds of markers, but for forensics, there are few markers. And there is a fun fact that if two persons, even for North and South India, when they meet, there are just 1.6% to 95% chances that they would understand each other's language. But you pick from anywhere in India, two persons, they would share more than 30% of their genes. So Indians are more connected with their genes even you compare any other population in the world. So if we look to the phylogeography of Y chromosome worldwide, we see that the populations like Africans, they have their own lineages, which is because Africa is the birthplace of the modern humans. So it can go up to the 140,000 to 150,000 years. And later there is a out of Africa migration. And then these regions, they have been populated by the people who came out of Africa. So there is like Indian specific markers are there, the European specific markers are there, Central Asian specific markers are there. So how to use these markers for the population genetics? Here just a basic information that to understand about the maternal ancestry, we just need the female line. The mitochondrial DNA is enough to understand the maternal ancestry. But for Y chromosome, it can give information about the male lines where we can get the information about the paternal ancestry. But what about the gray people? We don't have any information by using these two systems. So if we implement the autosomes, the biparental, as we all know, that 50% of our DNA we receive from father, 50% from mother. As we go back in the generation, we lose 50% of our DNA. So this is how, if we get the details of autosomes, we can answer about all our ancestors in the huge tree. And how we construct the haplotypes, how we make the trees out of it, and how we fit the individuals over the phylogeny. Here you see that there is a string of mutation, and this string of mutation is called as haplotype. And when these strings of mutations come together from several individuals, they are known as haplogroup. And when you design a tree, this tree, when there are bigger branches, they means they would have accumulated more mutation, so branches are bigger. When the branches are smaller, it means they might have less time to accumulate the mutation. So initially there was a paper by our collaborator. They showed that the initial Y chromosome after coming out of Africa actually differentiated well into the Asia, not into the Middle East and other region. So for example, like if we show that the African origin of Y chromosome, then it should show a differentiation from this region. But actually it was showing the basic branches are coming out from the Asian region. What does it mean? It means that Asia has worked as a reservoir for the modern humans, and the lineages has spread from Asia to other region of the world. Here you see that there are three haplogroups called haplogroup D, C and F, and these three haplogroups has very predominant presence into the Asian subcontinent. And then later we have compared the mitochondrial DNA and Y chromosome into the timeline of the modern human evolution. And here you could see that the Y chromosome, it shows a very clear-cut dip between 4 to 8,000 years ago. During that time, the ratio of a one person, one male was 16 female. So this was very high. Generally, it is 1:2. But why there is this kind of region? There we showed that it was, and initially when the result came out, I thought that this is because of Mahabharata. But Mahabharata has never happened into Africa. And this is a scene which has been seen somewhere, everywhere into the world. So what could be the region? We showed three kind of scenarios. One is Genghis Khan. You all know that Genghis Khan was a Mongol invader, and wherever he went, he just killed the males and then raped the women. And this is how there is 40 million people at the moment, 4 crore people, they are having Genghis Khan DNA. So this could be this kind of event which has reduced the Y chromosome diversity. Or when we started to live into the groups, there are king who could marry more. So one person could transform, could pass their DNA, their Y chromosome into more number of males. So this is how the Y chromosomal diversity has reduced during the 4 to 8,000 years ago. And there is one haplogroup in India that is called haplogroup H. It has been carried by more than 50% of the Indian male. This has origin into the southern part of India. Later, it has expanded into various regions. And here we have mapped the whole Y chromosome of haplogroup H. And you could see here that it has originated somewhere into the South India 24,000 years back. It has radiated to other parts of the Indian subcontinent. And then later there was a very special migration out of India that has happened something like 1,500 years back. And they are the European Roma, the European gypsies. They went out of India, and their genealogy has been reconstructed based on the Y chromosomal information coming from the YSTRs. And here you see that they are most closest to the northwestern Indian population. So some of the scheduled caste and scheduled tribe population of northwestern India, they went out of India and they are called as a gypsies. And here if we see the whole Indian map, you can see that with the color that there are few language families, they are present into India. So the Indo-European, mostly northern part, central part and western part. In southern part, there are Dravidians, and also into the Himalayan fringes, there are the Tibeto-Burman language families. And if we see to some of the tribal population, they are present into the pockets, into the central part of India and into the other regions. So there was another question that who are the Austroasiatic speakers who are present into the pockets? Many of them, they said that they are the one who came for the first time out of Africa. They are the one who arrived from out of Africa, and they are the one who first settled to the India. So here, when we look their Y chromosome and mitochondrial DNA diversity, here you see that the Southeast Asian population and South Asian population, they are showing two colors, and South Asian population, they are with the green color. And here you see that all of the Austroasiatic maternal ancestry is associated with South Asian. But if we look their Y chromosome, here you see that around 70% of the Austroasiatic ancestry is actually associated with Southeast Asia. So here is a story which shows that their maternal ancestry is indigenous to India, but their paternal ancestry is somewhere associated with Southeast Asia. How did we resolve these problems? We have used the autosomal DNA here because it says about all the information about their ancestors. And here you could see that there is Indian cline. Here is Indian cline. All the Indian, somehow it is not working. So all the Indian population, they are showing a particular cline, and this cline is present here. But the Austroasiatic people, they are here. And also there is another Khasi population, they are here. It means that they have gene flow from Indian population as well as from the Southeast Asian population. If they have 100% of Indian ancestry, they should have been lying here. But they are in between Southeast Asian and Indian population. And based on this, we have done their admixture analysis. In admixture analysis, one color shows one kind of ancestry. And here you could see the South Indians, they don't have the color which is present among the Austroasiatic population. The North Indians, they don't have this color. But the Indian Austroasiatic population and their neighboring populations, they are carrying this color. So it means that this ancestry actually came from Southeast Asia to India. So this is how the ancestral differences, the ancestry related populations could be differentiated based on their ancestry, and the timeline has showed that they might have migrated something like 4,000 years back from Southeast Asia to India. And here we could able to identify their 4 branches of ancestry, and these 4 branches were present into the various regions of India. And there was another haplogroup which is also indigenous that was present into various parts of India. And here you can see that this is the top sample is mine, the UP Brahmin, and there is a Kshatriya which is showing a common ancestry of 2000 years. Then they have both of these samples, they are showing a common ancestry with the Santhal of Jharkhand, something like 8,000 years. So you can imagine that how powerful these markers, they could show the differentiation of the population, the common ancestry of 2 individuals who have lived somewhere in the, like for example, like if we take the DNA samples sitting in this room, just do their Y chromosome, we can calculate exact the timeline that when these 2 people have shared the common ancestors. So by using these informations, we, and there is another question about haplogroup R1A. As you know, many of you know that R1A is actually, many people, they say that it has been associated with the migration of the people who is related with the Aryan ancestry. But based on our analysis, we showed that in all the branches of R1A, and these all the red dots, they are present among the Indian population. And these analysis showed that the Gangetic plain was the producer of around 95% of the R1A branches of this haplogroup. And here you see the distribution of this haplogroup is more towards the central India, not with the northwestern India. If you assume there is a migration, then you could see a gradient of those migration. And also we have done the Bangladeshi analysis, the population of Bangladesh, and we can clearly differentiate that the caste and tribe population of Bangladesh are quite distinct and quite different from each other. So the skeleton of tree is already ready. So if you have any unknown population, you could just analyze few of the markers. Any unknown sample, you could analyze just few of the markers, and based on their Y chromosome information, you could place them even among the whole world Y chromosomal tree. For example, like if you just discover a skeleton, something like 100, 200 years old, you can do their Y chromosome with the advanced technique, and you can precisely identify that this sample is belonging to which country, and based on our developed method, you can even compare with the different states of Indian population. And this is the information which brings about the whole plethora of the Indian Y chromosome. And here I bring another story which has just released few days back. There were 282 skeletons which have been found into a well in Ajnala, and this is in Amritsar, Punjab. No one knows that this belongs to whom. We have done their analysis related with the isotope analysis, their mitochondrial analysis. I was involved. This is the well actually. This is the well, and these were 282 skeletons. Who were they? No one knows. We have done their mitochondrial DNA, and based on mitochondrial DNA, we could able to clearly identify a close connection of these population with Gangetic plain population. So the people who have been died with the other historical information showed that they have been killed by the British people during the 1857 during the time when there was first freedom movement of India. And we have also done some of the analysis which has showed that these population are actually associated with the people of Uttar Pradesh, Bihar and West Bengal, and based on that one can say that there was 26th infantry which has been actually killed by the English people. So these information are being done based on the databases which has been produced by the population geneticists, and these population geneticists usually actually make the whole plethora of the landscape of the Indian population, and one forensic scientist could identify about the unknown population. So that's it. Thanks a lot. And I would like to acknowledge several people because we need to collect samples from several regions. Dr. Rakesh Tamang is here, who is one of the budding scientists and young scientists of India, who has helped us. Even he's the guy who has mapped the whole Himalayan region, and he's well known for his discovery of the Himalayan population genetics. So there are other people, Dr. Thangaraj, Dr. Lalji Singh, who has given us the knowledge to start to work in this area, Dr. Neeraj Rai, who has his own lab into the Birbal Sahani Institute, Professor George Von Reem, who is a linguist, but who is always connected with us, and we actually use his knowledge to explore our questions. So this is how a big, huge amount of people are involved, and thanks to the organization like ERC, National Geographic, ICMR, ICAR, and other Wellcome Trust, other agencies for funding us. So thank you very much. Please go for the lunch. Thank you so much.
Hello. Do you have any questions open now for questions? Any questions? Should be hungry for knowledge, for the purpose for which we are gathered. Sir,
Actually time is quite
okay.
so that's why.
Sir, I have one question, sir, to you. It's not an expert, an ordinary person. That there is an opinion, different opinions about the migration of the aliens, whether they have been actually migrated or they are the local people there.
Actually, sir, this is a question which is still under debate, under discussion. The problem is we lose ancient DNA from India. So there are some skeleton, we are still analyzing, but so far what we see that there are only 10% of ancestry of Indian population which is connected with people who were present into the steppe. So 90% of the Indian ancestry, 60% has been derived actually by the Harappan people, and 30% is coming from the indigenous people who are associated with the Andaman. So the discussion is about just 10% ancestry, but how this ancestry has come, still it is under debate, and we have to still analyze lot more sample to tell anything about it. But the Y chromosome which they have associated with it, haplogroup R1A, 95% of the haplogroup R1A is actually indigenous.
Okay, sir, thank you very much. One more question if I drag. There is some, the history says that there is a migration of civilization from one side to other sides, in spatial as well as
temporal
time.
Right.
Yes. And now, can Y chromosome study throw a light in reconstructing the history in terms of migration?
Definitely, sir, definitely, because you see, I presented the information about the gypsy population, and precisely we could tell that 1,400 years back, some northwestern population, they have gone to Europe, and this is actually quite correct.
Any such exhaustive work has been done in Indian context?
Yes, yes, actually it was started by Dr. Lalji Singh, and they had 25,000 repository into the CCMB, and we used those repository. So thanks to him, he was the pioneer who...
CCMB director.
Yes, yes, sir. Yeah.
Yes, I was actually very much fortunate when I was the director to have with him. And he, afterward that, he went to BHU as
yes,
a VC.
Yes, sir. Yes, sir. Yeah.
Okay. And he introduced the forensic science
there. Yeah, yeah. I teach forensic science there as well.
So I was known to him, but he's no more.
Yeah, yeah, yeah.
Sir, one more question.
No, he exists within us, sir. Yeah, please.
A lot of contribution. He was the father of DNA in
yes,
India.
Yes, 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 yes.
Establishing the fact.
Yeah.
We have great regards, endless love and regard to him. Sir, one more question. As you have studied this thing, what were the sample sizes?
Usually there are thousands of the samples. We collect samples from all the regions. That's why we collaborate many people.
Is there any standard method of selecting the sample sizes and the sample modeling distribution?
So usually, sir, it is random, because statistically, if we collect the random samples, usually we capture the whole area and whole landscape of the genetics. So this is the random, and we use all the three methods like autosomes, mitochondrial DNA, and Y chromosome. And using the three method, one could make a very holistic approach to answer any question.
Okay, sir. Thank you.
Thank you, sir.
Since there is no question from audience, we can conclude here, giving a big claps to both the distinguished lecturers sir, we are very much thankful. I hope that in future also NUJS will be benefited by your knowledge and experience. Thank you very much, Dr. Others.
Thank you, Gyaneshwar sir, for this wonderful session.